Shaoling,
Hey?
can you find out if Hua is going to come in?
Okay.
I think he is.
He's coming?
He said he was coming.
Okay. Trevor just went to his office.
Oh, okay. Awesome. Thank you.
Hello, this is Nansu.
Hi, Namsu. So maybe we just wait for 2 more minutes before we start.
Hi, this is Olivia Harris-Mendy, calling from
Hi, Olivia.
Hi.
Good.
We're just going to wait for a couple more minutes before we start.
And can you also ask Firat to give me more details about his problem with the account?
He's actually on the call, so maybe you guys can talk while we're waiting.
Yeah, that is great. So I'm not sure exactly what generated code I answered yesterday and I got the answer.
Hi.
What is the issue exactly?
I can connect to the VPN, but
Okay.
after that, with the RSA given to me, I can't log in with that.
Oh, so I see. So the RSA token is the issue.
Yeah.
Okay. I didn't know what exactly generated code. We have to be careful because the RSA application, if the pin is wrong, you'll still get the code.
Yeah.
The code is designed to be bad on purpose. So you have to make sure that the pin that you enter is correct.
The RSA version.
The pin.
The pin that you use for the RSA application. When you start the application, it asks you for a pin. If you put 1, 2, 3, 4, for instance, it will still give you a number, an 8-digit number, but that number will not be valid. So the pin has to be correct.
Yes, I use the pin that you sent to me. The
So,
4-digit pin, right?
Yes. And you're sure that is correct?
Yes.
Okay.
So I was able to connect, but then I lost connection. So once upon a time, I was able to connect.
Oh, I see.
Yeah.
Okay, at least if the VPN is working, that's a good sign. I will get Daniel, and he will contact you with regard to your RSA pin, and he'll probably have to reset it. Okay?
Thanks.
Okay.
Okay, so maybe we'll get started. Right now, before going over the agenda, I just want to talk about who's all on the call. So on the call, we have Claudio. You already heard him speaking. We have Elizabeth, Jeff, Jihoon, Olivia, Nansu, and on the UTH side, we have Shaoling, Firat, Trevor. Did I miss anybody?
I'm here too. Wow.
Hua.
And I have the postdoc who did the work with me in my office. His name is Max.
Hi.
Max. Okay. Let's get started. On the agenda today, we're going to just talk briefly about updates on action items, and then I want to briefly touch on the version 3 testing plans and release plans for DataMesh. I don't know where it went missing. And then Olivier is going to talk about the deployed supplement and how we plan to integrate it. And then maybe he's going to present the supplement work, and then we talk about how we will integrate it into DataMesh and also the timeline of this integration. So for action items, currently we have the data type ontology that Philip and Alejandra have already done the first pass with, and it's available on GitHub under the BioCaddy, under the BioCaddy institution. And then you have a repository which is called RAO. And so, as mentioned before, please do go in and review, because I think they're waiting to get some feedback from us. We also have the JMIA review. So I put up both the reviewers' comments. I think Fa has gone over those and suggested and put in his comments also in addition to the reviewers' comments. And I also put up the submitted document that we had. So that's in the same Google Doc. I can send out an email after this meeting with links to both again. So please do go in. I know Burak has put in some information already, and Xiaoling and I just talked briefly. We will start working on it probably after tomorrow. So the plan is that, so it has to go back on the 5th of August, and so I'm hoping that we can get at least our updates done by the end of, or at least mid next week, so that we can then circulate it to the other members and get their feedback. So for the document testing.
This is Julie. I just joined. I wasn't sure if I was expected here today or no.
I don't know that we are going to talk about the supplement, about your supplement today, though, Julie. I mean, you are welcome to stay on if you would like to.
To be honest, I've got several fires concurrently, so if it's all the same, I think I'll bow out. But do let me know if anything comes up. I'm available on Skype if you want me to patch back in.
Sure. Okay.
Thanks.
Thanks.
Okay. Bye-bye.
Thanks.
Bye. So for the DataMed, testing began on July 17th, so basically yesterday. And I know that we've had some activity already with the folks that are going in and testing. Basically, as we did earlier, we're tracking all our testing and providing comments on Trello. And the test site is at datamedbeta.biocaddy.org, for which, in order to use it, you need the username and password. So please do use that and to get into it. And the testing is expected to be completed by Thursday evening, so that Firat and Xiaoling, as well as Burak, if there are any issues with the other part, although the testing is primarily for the UI at this stage, will have some time to complete the test or fix any bugs. As mentioned earlier, we also talked about the release, and the release is going to be with a release announcement sent over to a list that was previously compiled. And we will also send a release announcement to the BD2K for the newsletter. And if any of you are part of mailing list, please do let me know so I can send you a copy of the release announcement, and that could be put into the mailing list as well. I guess that's about the general business for the CDT today. Now we'll have Olivier present the deployed supplement, which was a supplement to the BioCaddy project. And after his presentation, I think we would need to discuss how and we'll be integrating it into DataMed. And the primary thing to remember is that Lucilla suggested that we should try and get it integrated before the all-hands meeting so that it can be demoed at the all-hands meeting as part of DataMed. I do not know if this timeline is possible, so maybe we would need to talk about that as well. Olivier, please go ahead.
Okay, so I sent a few slides. I don't know if people received them. I'm not sure how many people were on that email. But if you have them, just open them. I'll walk you through it. It's a short summary of what I presented in the BioCADI webinar series last week on the work Max, Ji-Hoon, and I have been doing for the past couple of months. So on slide number 2, we can explain the rationale. We want to add a piece of metadata to some of the dbGaP cohort, pretty much. And this piece of metadata, we call it the diversity index, in a way, which inform us about the racial and ethnic diversity of the different dbGaP cohort indexed by DataMed. There's a strong rationale for it. We know there's a lack of diversity in most of the cohorts that are being assembled for biomedical research, and we want to be able to illustrate that better, and we want to be able, for researchers using this public data, to be able to select a cohort based on the admixture and based on the race and ethnicity, going beyond the self-reporting information that's sometimes included in these cohorts. So the goal, again, is on slide number 2, is to include the diversity score for multiple cohorts indexed by BioCADI and DataMed search index, and with the potential utility that I just described. So for this, on slide number 3, I illustrate what we rely on. A number of these cohorts have genetic information available, either under the form of the results of the genotyping array, of an exome sequencing, of an RNA sequencing. Therefore, it gives us access to genetic variants. And we know from the 1000 Genome Project, that is illustrated in this slide, we know the genetic diversity of a number of populations around the world that we can group into 5 superpopulations. You can see a superpopulation in East Asia composed of 5 populations in green, South Asia, Africa, South America, or American, Native American, let's say, and then European. So those are the 5 populations that are further illustrated in the slide number 4. And so there's like 25 populations that have been collapsed into 5 superpopulations. And so the 1000 Genome Project generated all the genetic information from this population for a total of 81 million variants. So this is the reference data set. So this is our reference, and this is where we're going to compare the variants we find in each cohort to this reference panel. And using a method to call admixture. And this is, on slide number 5, an example of the result of an admixture calling, for example. You can see the genome of 2 people, one on the left and one on the right, either at the global level, represented by this pie chart, or at the chromosomal level, represented by all those colorful segments. And those 2 genomes are from people from Colombia, and you can see that they vary differently, very differently, based on their ancestry. For example, the person on the left has about 30% of 3 different ancestries, 30% African, 30% Native American, 30% European. In contrast, the person on the right is mostly a mix of Native American and European with very little of African ancestry. So this is what admixture mapping allows us to do. I mean, with very high resolution, it can really allow us to pick different segments on chromosome. This is not the goal of Diploid. The goal of Diploid is the pie chart that you see above, okay? And it's even actually a broader goal. I'm going to explain it. But we're going to stay at the very high level. So for this, we selected a number of studies from dbGaP on which we thought we could do that would allow us to go dig inside the data, call the variants, call the ancestry. And so here, this list of cohorts is to illustrate in this very colorful column on the right what data type is available from this cohort. The best cohort we have is the one on top. Is called the TCGA cohort, actually the 2 ones on top, the TCGA cohort. Those are great because they have all 4 data types, either a SNP array or a whole exome or whole genome or RNA. So we can actually use those to kind of refine our method to call admixture and to make sure we have a robust method that is kind of agnostic to the data type. But in general, the vast majority of the cohorts have either the SNP, which is the red column, or the whole exome, which is the blue column. So in slide number 7, I explain different methods for the different data type. You can see the main method is the one in the middle, which we start from the result of a genotyping array. We call genotypes across the entire genome, about 1 million genotypes per sample. And then we use this gray population reference from the 1,000 genome to call admixture. We call admixture first at the individual subject level. So every person in the cohort is going to get 5 values pretty much from those 5 superpopulations. So it's a vector of 5, size 5. And then we just accumulate, we sum up across the entire cohort those 5 components. So we get a cohort-wide admixture. And finally, the last number is a summary index of the admixture, which we call the entropy. So when you look very carefully at this process, really what we are interested in putting into DataMed are the last 2 black boxes, okay? A cohort-level admixture, which is a vector of size 5 of each component, European, African, East Asian, South Asian, and American. And the last number, the last box, which is the what we call the entropy. And so I have a couple examples in the following slide that illustrate this. We did that on the TCGA cohort, which was more than 10,000 different patients, and you can see that we can break down either the entire cohort in the upper left corner, and you can see that in total it's almost 75% European and some African, East Asian, South Asian, and American. But you can also make sub-cohorts. Oh, there's a barking. We can also make sub-cohorts from these cohorts. And you can see that they're pretty boring. They're all pretty similar. The breast cancer may be the most diverse one with more African. And you can see that this E number is the entropy number, which is indeed for the breast cohort the higher. So it's a number between 0 and 1, 1 being the most diverse. For example, if you had, 20% of each of the 5 populations, and 0 between the least diverse, like if it was totally uniform of one superpopulation. I present another case in the next slide, in slide number 9, where at this time I broke down the TCGA cohort based on the self-reported ancestry. So we had the self-reported Asian, and indeed the vast majority of the diversity of this cohort comes from the East Asian ancestry, and so there's very little entropy, 0 to 35. In contrast, in the lower right corner, you can see the people who self-reported themselves as Hispanic, and there's a fair balance between European and Native American, which is what we expect, and this one has a much higher diversity index of 75. So again, for each cohort, we have pretty much 6 numbers to put in DataMed. 5 numbers that correspond to the fraction of the 5 ancestry, and 1 summary number which corresponds to the entropy of the index. So in the next slide, I describe what you guys may be the most familiar with, which is this beautiful GUI that you guys made for searching and sorting this dbGaP cohort. When you are in dbGaP, you have additional metadata that you can filter on, like disease or study type. That's on the left column. And what I propose is to add another one, so that's in slide number 11, to add another one, which is allow us to filter cohorts based on the summary index, the minimum diversity that I call, from very low diversity, which is 0, to very high diversity, which is 1. And in addition, because diversity is not enough, we would like to know what is the dominant ancestry in each of these cohorts. We can let the user select, okay, I want main ancestry African, and I want my cohort to have more than 50% African, for example. So this is just a proposal. You guys are much more familiar on how to make it user-friendly and painless, but this is what I wanted to present. And so the conclusion of this project was that we believe that this ancestry is a critical covariate in most genetic studies. So we believe that it would be really an additional, very important metadata to add to the dbGaP cohort indexed by DataMed, because we cannot rely on self-reported ancestry. And we found a simple way to summarize this diversity through an entropy number as well as the major ancestry that are in the cohort. And we would love if we could work with you guys to put it together. So these are the people that were involved in the project, supported by Lucila Ono-Machado, and Max and Jihu did most of the work. So now I'm ready to hear your feedback and let me know how hard do you think it could be.
I think it's not just like how hard it will. It's more like I think it's doable. It's more about how much work it will be. Can we actually finish before the all hands meeting, which is when is it? August 7th. So my feeling is something on the back end needs to be changed, right? The database for individual repository to store this kind of information. Then the other thing is on the interface side.
So there's no quantitative information in the database currently?
Say it again?
There's no quantitative information in the database currently? It's only like flags or something?
No.
Yeah, I mean, there's no quantitative information about the actual diversity numbers you're talking about. So it's I mean, that would need to be added.
Jeff, do we need to change adding any database fields to store those information on?
Yeah, I mean, we need to add. I mean, there need to be sort of metadata attributes that store that information, right? And then off of that, you could derive various metrics.
So, but for now, Olivier, can I ask, you've done this only for dbGaP or only for TCGA, or how many of the repositories that are represented in DataMed do you have this information?
Yeah.
We've done it for TCGA right now. And that's what Dr. Ono-Machado suggested. You know, we stopped there just as a demo. I understand that it's more involved because if you put something, it needs to be generalizable to everything. But right now we only have it for TCGA.
And you already have an index created with the numbers.
Yeah, it's pretty much
all the data sets in TCGA, right?
So the thing is that the TCGA data we have, I don't know how it relates to the dbGaP study that you index. It's always a bit confusing to me. If you look at dbGaP, TCGA, I think there's two projects. One has 5,000 participants, one has 300 or something. I think I have them in mind. But if you actually download the data from TCGA, you get data for 10,000. And it's unclear to me. I guess we should say it's the one on top of the page, slide number 6, where it's PHS 00178. That would be the one that we relate to.
So since you've done TCGA, actually, I'm wondering if we should be just not, instead of going to dbGaP, we would just include this information as an additional or an outside index, like we did for PDB-HF. Is that possible?
Like we did for what?
So for PDB, for all...
PDB, okay.
Yeah, for the literature linkage, what we did, or for citation, what we did was we had information that was given to us by CETA group, and that was provided as a separate index. And Jeff was able to pull that in.
Yeah.
Right?
I mean, we have the data for that. Yeah, I mean, we could merge the data from from that supplementary source. The question is what are the specific fields and what would we need to store internally in the index to, what are the additional, I mean, what's basically the additional information that we need to extract?
I'm going to give you 6 fields, 6 numbers, and that's it.
So we don't need to extract any.
You don't need to calculate anything. You don't need to extract anything. You don't need to run any pipeline. We do that. I provide 6 numbers.
Right.
And then 6 numbers associated to then a data
one
set.
study. 6 numbers per study.
So most likely we can put this into the the dimension field maybe. I don't know. If we don't want to add like additional fixed field.
So I would
What do you think, Jeff?
So at what level? Can you see my screen now?
Yes.
Does it show DataMed?
Yeah.
Okay, so at what level do you have it indexed? Because TCGA has it at there are various levels that it has data at, right?
Yes,
so
that's my question too. Basically, the way how many data set we have about TCGA versus how many data set Olivia have about TCGA. Why they are different? That could be because it's on a different granularity level. First, we have to make sure we are actually on the same level. Then those additional metadata can easily add here. So how many TCGA data set we have right now, and how many in their database?
So if you see DataMed, I've just pulled up TCGA that we have information that we have currently from DataMed. So we are indexing at a very high level. This is how many we have, and we are indexing at the level of the project actually.
Yeah.
So let's say we go, and then if you go into each one of them, that's all we do. We just direct them then to TCGA because we are really indexing at a very high level. So we do not have metadata that goes deeper into not even like individual level. I mean, I presume you would go to patient level, right? For your work.
No, on the contrary, what I described that we're not allowed actually to share that patient level information, right?
They're also on the study level, but I don't know, is this study level same as the project level we have?
So that's what I'm not familiar with. I'm looking at DataMed now. You guys have a bunch of things that are indexed that are labeled TCGA that I'm not necessarily familiar with because that's not how I get my data. I see proteome thing. So I guess every sub-project has a different set of patients in it. Mm-hmm. And that's not what we've done. And so that the connection between your list of projects and our work is not easy. Like, is there any way to know which patients are in which project?
Not based on what we have indexed right now, no.
Because I have this for all TCGA patients, all the union of all projects, but you guys don't really index that.
So when you say TCGA, you're talking only about TCGA, or are you talking about GDC, Genomic Data Commons? Because
Genomic Data Commons has more than TCGA, right? They also have the target trial. They also have a bunch of other things. No, well, we definitely use the GDC to access the data, but we only.
I think it will be helpful maybe send the data description you have, and maybe Jeff, if your team can take a look to really figure out what's the difference in terms of granularity.
So the date. So the level. So do you have a notion of what projects the data is associated with that you have, or?
Yeah, because I know what project I got authorized access for because I applied to dbGaP to download this data, right? So in my slides, the slide number 6, the top 2, projects are the ones that give you access to all TCGA data, pretty much.
I think we can generate list from what he has and then look at the list we have, then we can actually find something. That's actually really important. Otherwise, if it's not really on the same level, we cannot just easily use all the numbers generated already. So
it's very so if we find a subset of samples, it's very easy for us to generate those numbers again for a subset of samples. But that's why I was asking, do you have a way to know?
But I don't think we have those individual patient level for each data set we have in the DataMed.
So okay, when I type TCGA in DataMed, the number 2 thing that comes up is a very general one. It's Homo sapiens TCGA Cancer Genome Atlas Bioproject.
Yeah.
So how about we say that's this one? How about we say that what we studied and what we generated this diversity index is for this one?
What do you mean by this one? I don't follow what you're saying.
There is a project.
So when you're saying TCGA, right, you're talking about all different cancer types
Yes.
together, correct?
Yes.
So do you have information? Because we have ingested TCGA data for each specific cancer type.
Okay.
So if you have the information, or if you can generate information for TCGA for those specific cancer types, I guess then we should be able to use that information.
This is my slide number 8, right? That's what I did. Yes. So it's very easy for us to slice the data the way you want.
So you mean this one, the one which talks about the cumulative fraction by disease?
Yes.
Okay. So
is this the level for DataMed, how we ingest TCGA data by disease? Each data set is a disease?
Yeah. So you can see, I don't know if you're able to see my screen.
No, I'm not. I'm not on the web conference. I'm only on the phone.
Oh, okay. So if the others can see the screen, so for TCGA data that we have here, I just pulled up GBC, but even within that, you can see for the TCGA, it's by diseases that we have. So
in that case, if those two levels match, then we can display those at the disease level, basically. And yeah.
But I mean that's going to be hard because, for example, there's one that you have that says proteomic analysis of TCGA colon and rectal carcinoma. But I'm pretty sure that not all colorectal cancer patients in TCGA had the proteomic done. So in order to be accurate, I would need to find out, okay, what are the patients in that thing you index? That's really hard.
Just another question. So I do not see the proteomic part that you're talking about.
I just typed TCGA in the search bar, and that's all I did.
Oh, okay. So that would pull up things from other repositories as well.
So which repository do you recommend I use?
So you would need to, in order to look at what...
On the left side, only select the TCGA as the repository, then it shows all the results from TCGA. But if you search on the search bar, say TCGA, then it may involve other database which also use TCGA sample but generate other proteomic data or other data.
Okay. Let me look for the the TCGA repository. I don't
So
see
it's
it.
on the left. If you limit the repository to TCGA only, then that's what you will see the data set.
I see dbGaP, but I don't see TCGA.
It will be called GDC because we actually consumed metadata from the genomic data
Oh,
commons.
GDC. Okay, I did GDC. Okay, I have GDC. All right. All right. But GDC has more than TCGA in general. But okay.
Right,
That's
but
right.
Okay, got it. All right, perfect. Okay, so that may be better. Yeah, okay. So we can rely on the we do know which patient is from what disease. So like right now I see 39 different GDC projects. So
Yeah,
I can give you 39 times the 6 numbers. You want that?
Yeah, maybe that's the easiest way, I think.
Let's do that.
Jeff, is that enough?
That'd be perfect for the demo that Lucila wants, you know.
I think so.
Jeff, do you think that's
That
enough?
will probably not much work. Otherwise, the other better solution is we have to ingesting TCGA or genomic GDC at a much more granular level. So we may want to take all the data set from Olivia and re-ingestion them.
But is that the question then is that really then a data set, right? What is the level of the data set? And that's some of the questions that I have for later topics as well around things like BioGrid.
My question is also really when the user trying to grab a data set from TCGA, are they really looking at this disease level or they're looking at the fine level? So I think it all depends on the user.
So initially for TCGA, actually, we had indexed at a very, very granular level, which was really at the level of individual files for each patient. In TCGA, each patient has multiple analysis done on their on their samples, right? So we were at that stage, and then we had a couple of discussions with Ian, who suggested that this might be the more appropriate granular level for injection, at least initially, by DataMed.
Yeah, because you don't want to be redundant with the GDC. I mean, the GDC already performs a number of indexing and dashboard at the individual patient level.
Right. So, which is why we, I think, re-indexed it at a higher level.
And the data I generate really has not much value yet at the individual file level. I mean, in the spirit of the supplement, we really wanted to provide a summary at the cohort level. Now, I understand the TCGA is not really a cohort, and that the way you guys thought about TCGA was by disease, and that's perfectly fine with me. And I'm happy to say we did it for 39 cohorts. It's okay.
So Jeff, if we get those numbers from Olivia, can we then quickly add that into just the TCGA that we have here?
We should be able to.
And then we could have an additional, I guess, a supplementary bar here, which shows some kind of, but we would need a little bit of explanation of what it really is.
Yeah, we can provide documentation on how that was generated.
Yeah, I think the easiest way is let's put those 39 data sets and add to the demo for now. Then we decide what to do later.
Okay, that sounds good then. So Olivier, can you generate it for these specific based on the diseases? I mean, here for GDC we have both target and TCGA. you could just pick the TCGA here that we have.
Yeah, I didn't do it on target, but I actually I don't see the target ones, so they're not showing up. It looks like you're on yours. But yeah, I would provide, a matrix of 39 by 6 or something like that, numbers.
Jeff, once we get those 6 fields from the Olivia, can you put it into the database somewhere?
Yeah. I mean, once we have that data, then Barak will just have to write a little bit of code to inject that into the data records.
Okay. We'll just work on the interface them, right? Just to
Yeah.
assume there's something in the database. As long as you let us know, then we can start working on the interface.
And then if that information is there as one of the metadata, I think Xiaoling should be able to select that as a filter, right? Xiaoling?
Yeah. So we can define like the range filter or the selection filter.
So on the UI side, it should probably not be very hard to integrate it.
Yeah, maybe 1 or 2 days.
So do you think it's doable before the all hands meeting then? So Olivia, how long do you need to be able to generate the numbers for us?
I'll send it tonight.
Oh, okay. So Jeff?
I think it's okay. Sounds like we can do it.
Okay.
Excellent. Great to hear.
So Oliver, if you can send it to the same people who are on the email thread, or just to Jeff, it would be great. Or if you don't have Jeff's email, you can send it to me and I can forward it to him.
I will.
Okay. Sounds good then. Okay. So is there anything else?
So Jeff, do you still have like new repository coming for the version 3.0?
So there were a couple that Barak sent over. There are some from that we need to talk about, like BioGrid, and then like the Omics DI is being split. Barak's, I was at NIH yesterday, so I need to check with him, but I think he might have that code almost done, so we can do that. But then there were a couple like BioGrid and others. There were a few where it was hard to know if we were actually indexing data sets or what was the data set. So, but I don't know if we're in that section of the agenda right now, or.
We can talk about it. Jeff, do you want me to make you a presenter?
But don't need anything to present.
Okay.
So, for example, MGI. So MGI has more than 24 entries in terms of transformations, and so the question is, for MGI, what is the data set that we're looking at trying to index, and with BioGrid it's a similar issue where we have, in a sense, it looks like we're indexing the actual interactions, like individual interactions, and I'm not sure if that would be considered a data set.
Oh, okay.
Because
so these were for the updates, not for new repositories, right?
No, these are new. So MGI is new. BioGrid is new. And so for some of these, I mean, BioGrid is sort of like Unipro, right? In that it has a list of information entities. But unlike Unipro, each information artifact is very granular, right? It's an individual interaction. And does that make sense?
No, I'm just going to share the screen because I just opened up the transformations. For BioGrid, I see there are like 1, 2,
yeah, I know exactly.
3,
Yeah.
4, 5.
Yep.
5 different transformations.
And they're
Oh, okay. I haven't looked at this, so I will. Protein chemical interaction. So is there like a higher level that we should maybe look at?
So
for BioGrid?
Well, I mean, the question is, how do we want to break down? Because they do have, so go to downloads. Yeah. I mean, basically the BioGrid is a single data set, right? There's a current release.
Sample data.
They have some.
Oh, they
So
have.
go to the BioGrid website, right?
Yeah, so I'm just trying to go to that. So by gene.
And so if you go to go to downloads to the right.
Okay.
So they have you found current data set, right? So they have the current data set, which is sort of the BioGrid all, right? And then they do something where they split data sets into different files based on organism, based on chemical, things like that. So the question is for because the BioGrid data set is basically these, these interactions. So the real question is, how do we want to reference that, right? Because we could potentially, split them up so there are 1, 2, 3, 4, 5, broken down by organism. So then at least you'd be able, then you'd have, the data sets by organism. But it's actually, there are more organisms than just the 5. So, it's really, how do we want to slice and dice these informational data sets, right? So, because it is a data set, and at what level do we want to display them? So
what's the system.
Pardon me?
There's something that they've split it as system. So each of these, for example, if it says organisms, it has 1, 2, 3, 4, 5, right?
Yeah, but if you go to the, there's a, where do you get this? The statistics is under stats. Oops. So go under stats on the top. And so, there's a number of, there's interaction based on species, right, which is the top one.
So is it possible for us to just get to all of them? I mean, if you go to this and go to all.
Yeah, all would give you all of that. So the question is,
and then we map it from there? Because do they have like cross-species interaction studies?
I don't know I don't think you can have cross-species interactions, right? Because it's chemical interactions and other interactions that are done in, various preparations. So it would have to be within, a certain model organism that they're testing something.
Oh,
right?
It's that way. Okay.
Yeah.
So then maybe based on, is that possible that we just download all and then try and map?
Well, that's so we do have one part of the data which lists all the interactions, right?
Mm-hmm.
I believe, or is it ChemTab? Go back. Yes. So go into go into that. So you were at PTM. Yeah. Go down into. Just choose one of the records.
But this is just, so I am assuming PTM is basically post-translational modification, right?
Yeah, no, exactly. But if you look at the records, right, it's not. It's not really a data set. Again, it's a piece of information. So the question is do we count this as a data set or not? Because if it is a data set, then we have to sort of construct, a name around it of what we. But again, it's one piece of. This is the data, right? So if this is the data set, this is the data of that data set. Or do we bounce up a level? It gets to the scoping of the data set issue. Because this basically is one, one fact.
Yeah, this is one gene, gene and one PTM on that gene, right?
Yep, extracted from one publication.
Okay.
So I don't. Yeah, again, the question is, how do we want to? What is the way that we want to again slice and dice that data set?
Oh, I don't know. Probably, I guess we need to look into it a little bit more.
Because it could be. It could be that it's just the, that we just sort of index the high-level data sets, with the organisms listed, for example. Right. So again, it's again just these interactions between, in this case, genes.
Okay, so this is. I just think we might have to move up. Is there are there different ones based on publication? Can we go at the level of publication if that's possible?
Yeah, I think that's possible.
Potentially.
Featuring.
No, but those are publications on the data, I think. So you're talking about the publication that they extracted the
data from
around.
Right. That would be basically the primary publication that produced that data set. I mean, that the data set produced.
Right. That might be possible, yeah.
But it looks like they have publication information.
What we can do is we can look at that real quick and then, do a sample of what that might look like.
Okay.
Okay, and then we can have a to show what that is.
Okay.
All right. Do we have anything else?
No. So the only other thing is with regard to the opening up of the GitHub.
Oh yeah. Make sure we talk to Lucila about this. So, Jeff, I'm thinking about for the data meta paper to basically to address the reviewers' comments. I try to make the paper sound like we developed this data meta and there are actually a lot of needs for people trying to build this kind of index. So another contribution of this is a framework for doing this kind of index. And
right.
we want to say we provide this open source packages on the GitHub. So eventually we're probably going to open up the GitHub for other people to be able to download data map framework to build indexes. So I think this will be a big contribution, and we can sort of convince the reviewer this is not just about why we're doing these technical details, because we are providing this package and the people want to learn the details so they can reuse this package.
Right.
So I need to talk to Lucila about this, open up this GitHub. But I think she's probably aware of it because at the beginning we always say this is an open source project and we will open it.
Yeah, and I mean, there's some of the code already sort of in open repos, right?
Mm-hmm.
Yeah.
Some of it needs to be synced.
I told Anu is we probably don't have to organize everything very clear, very well before we open it. We can open some of them, and when people starting download, they have questions, we maybe help them to get. Then we get organized the GitHub as well.
Right.
Yeah. Okay. All right.
So just to make sure that there's no login information or anything in the code, as long as we don't have any of such information, I think we should be fine,
yep.
Right?
Mm-hmm. All right. Okay. I think we're good. Thanks, everyone.
Thank you.
Bye.
Thanks all.
Bye.
Bye.
You have terminal? You have terminal.
From your terminal.